Hi everyone, my name is Blessing Awabmiller, and I'm working alongside Paula, Goody, Rosemary, and Jenna on a project regarding Arabidopsis thaliana.
So drought is a major agriculture and economical problem. Specifically, its effect to the crop yield. As per report, by 2013, 40% of the world population will be affected by drought condition. So find a better solution. Here we're testing the mitigation of a drought on specifically dicot plant Arabidopsis thaliana by applying rock or honey to it.
Biostimulants, such as honey, are any microorganism or substance that enhance crop quality traits, increase the efficacy of nutrient uptake, and can enhance tolerance against abiotic stresses, such as drought. However, artificially made biostimulants can be expensive.
Honey has a lot of beneficial components that make it a good candidate for the purposes of this study.
There have been previous studies testing honey on onions and chili peppers. However, they are not model organisms, and honey has not been tested on Arabidopsis, which leads to our hypothesis that honey can mitigate drought stress in Arabidopsis thaliana.
To test this hypothesis, we divided two groups, one being a group experiencing drought conditions and another being a group that experienced normal conditions into four different subgroups. And two of these groups were treated with concentrations of diluted bee honey, and the results that we gathered were very significant.
Hello, everyone. My name is Alex Angel Nunez. I am going to be discussing my search for adenovirus within Cuban nitinols in southern Florida. ADV, also known as adenovirus, is a DNA infection that infects most living vertebrates. The virus I'm mainly focused on is adenovirus. It's an adenovirus genus that affects mostly reptilian hosts and can cause severe symptoms and death within the host. It's thought to co-evolve with hosts and horizontally transfer, which has been shown in recent studies. There have been no known studies of adenovirus and its effects on Cuban nitinols. And for my methods, I extracted DNA samples from 8 wild Cuban nitinols, and we performed nested PCR utilizing ADV-specific primers, and we used those PCRs to test for ADV infections within the samples, along with amplifying the DNA. And we performed gel electrophoresis to confirm presence of ADV within the samples. With this, we were able to detect one possible infection of Cuban nitinol, which would be the first ever identified.
Hi everyone, my name is Eamon and I'm visualizing recurrent structural variants that are affecting tumor genomes using GRAM and VN. So cancer has affected millions of people around the world, and although we currently have some developed techniques for its treatment, they have their limitations. Hence, a different approach must be studied upon. The approach we're looking more in depth is identifying the structural variants responsible for tumor development and allowing other researchers to target genes associated with them and open more doors for treatment. This project is fully computational, and we're using two main computational tools along with the whole genome sequencing data of multiple myeloma patients. Stop by my poster to see the genes identified and their functions.
My name is Dier Kaboski. My project is titled The Functional Characterization of SBTs in Arabidopsis thaliana. During this project, we identified two subtalases, or SBTs, which are peptidases that have influence over stomatoformation and vascular development in plants. The data I have today includes a PCR experiment to confirm the presence of the tDNA insertion that caused the SBT loss of function mutation in the plants, a root growth experiment, and a stomatophenotype experiment. The most insightful results were obtained from the root growth experiment, which shows that both SBT lines have significantly longer roots than the wild type. Because of the results of this experiment, we plan to look further into these SBTs' influence on vascular development with more growth experiments to understand the mechanistic role of these SBTs.
Hi, my name is Elizabeth, and this is my pilot study, which explores the dietary monosodium glutamate's effects on nociception in Drosophila melanogaster. I was fortunate enough to work with Dr. Nathan Fried over the summer, and I relayed to him my interest in nutrition science, and his was in chronic pain and neuroscience. So we married both of our interests, and I decided to use MSG, or monosodium glutamate, because it was the perfect culprit in the dietary aspect of how pain can be formulated or as a factor of pain. So we know that pain can come from injury, sleep, illness, and like I said, diet. So we view this using Drosophila melanogaster, specifically in its third and star state, to test the latency across the 45 milligram MSG ingested third and star larva and my negative control of 0 milligrams. we were able to see a slight significant trend in which I will be increasing my sample size from 15 to 32. After running a power analysis, I was able to see that with the increase of 32, I will prevent a type 2 error and continue to use my 10% hydrochloric acid, it being the LD50 of this experiment. We just focused essentially on the upregulation of AMPA receptors on the synaptical cleft of these glutamatergic neurons and hope to see more of a change with the increase of concentrations. If you want to learn more about my project, hop into my Zoom room. Thank you.
Hello, I'm Isabella, and I'm a student in Dr. Nathan Freed's lab. In his lab, we study mechanisms associated with chronic pain. We want to further understand what specific mechanisms determine variability levels in pain and how it relates to sleep. To fully characterize this clinical phenomenon of pain in humans, we need to characterize this in Drosophila first. A thermal assay is one behavioral assay we can use to measure pain. My project focuses on adopting a tool to measure thermal sensitivity, which allowed me to conduct multiple trials and produce data. I found variability levels and latency observed in nociceptive behaviors. Characterizing thermal nociception behavior in Drosophila larvae to study the impact of sleep disruption on pain is only the beginning to unlocking what molecular mechanisms are involved in the clinical phenomenon of what we know in field of pain. If you are interested in learning more about my project, please visit my Zoom room. Thank you.
Hello, everyone. My name is John Crespo. I am currently a senior at Rutgers Camden, majoring in biology. This summer, I had the experience of working on a cancer immunology project at the University of Pennsylvania. This project consisted of exploring ways to target the limitations of chimeric antigen receptor, in other words, CAR T-cell therapy, which is a form of immunotherapy that primarily treats hematological malignancies, or in other words, blood cancers. This consisted of modifying the elements of recognition in the CAR and utilizing a lentivirus to bring in vitro work to in vivo by selectively transducing CD8 plus T cells. By visualizing in vitro transduction, we saw a selective CD8 T cell transduction across a T cell expansion and a titration, indicating that these two modifications of targeting the limitations of CAR are heading towards the right direction in targeting limitations of this immunotherapy.
Hi, my name is Blessing Wabamila, and I worked alongside Dr. Kwanwan Lee on this experiment regarding photoperiodism in Neurospora. So to measure photoperiodism in Neurospora crassa, which photoperiodism is the response of an organism to seasonal or annual changes in their environment, we compared 3 strains. One being the wild type, we compared that to 2 knockout mutants, and what we compared was the average number of photoperiodicea produced, which is the female sexual structure produced in Neurospora. And this is a great unit for measuring photoperiodism in Neurospora. So once we collected the photoperiodicea produced in Neurospora, we compared these with the different strains in different photoperiodic conditions, which were various amounts of light and darkness, to see if there's a statistical difference.
Hi, my name is Rosemary Arroyo, and I work in Dr. Freed's lab on mechanical stimuli and how it affects Drosophila larvae. 1 in 5 people experience chronic pain, which leads to an impact being made on their daily lives. Daily activities such as walking and working can become a challenge. In this lab, we are adopting a technique to test von Frey hairs on Drosophila larvae. Von Frey hairs are synthetic fibers used in pain research and are perfect to use on a model organism like Drosophila. Drosophila end up being an ideal organism for testing several nociceptions, such as mechanical stimuli, thermal stimuli, and chemical stimuli because of their small size and their ability to reproduce quickly. In this experiment, we are going to adopt the technique so that it can be used in the future for a better understanding of how mechanical pain works.
Hi everyone, my name is Eamon and I'm studying the effects of global change drivers on the susceptibility of vital plants to pathogenic diseases. As you guys know, human activities have intensified over the last 200 years, resulting in major environmental changes, which are negatively affecting the Earth's ecosystems. These drivers have been shown to severely affect plant and pathogen interactions and the extent of disease they cause. These diseases are predicted to disrupt the functioning of ecosystems and their services to human populations. What we're actually asking is, how do these drivers affect the susceptibility and severity of diseases caused by bacteria, fungi, or viruses in wild plants? To answer this question, we performed a meta-analysis, and to see my results, stop by my poster.
Chronic pain is a worldwide health concern that affects many people. As of 2022, up to 40% of Americans suffer from this condition. Historically, opioids have been used to combat pain, but that also comes with many side effects as seen with the opioid epidemic that we are currently battling. Due to this, there should be many other ways that come with less severe side effects to help treat chronic pain. My project is focused on trying to understand more about how chronic pain works and if pain levels change throughout the time of day. I'm using the animal model Drosophila melanogaster to understand its correlation with the goal of providing future treatment to those burdened with chronic pain, especially people who work the graveyard shift.
As a group, we are studying Drosophila melanogaster, a common fruit fly, and their seizure susceptibility. We are focusing on their response to chronic pain caused by leg amputation. This specific injury triggers a loss in GABAergic neurons responsible for inhibiting the overexcitation in the brain. Increased excitation in the brain can be presented in patients as epilepsy. It is presumed that a loss of these neurons will overexcite the brain, causing an increase in seizure susceptibility.
We embarked on the study to further improve our clinical knowledge of seizure-causing syndromes and connect the effect of chronic pain and seizures. Additionally, we have discovered that patients with chronic pain conditions can experience non-epileptic seizures. We are interested in testing this in order to have a greater understanding of the multiple triggers that are associated with epilepsy and seizures, both epileptic and non-epileptic.
Hi everyone, my name is Shweta Samuel, and I'm going to be discussing my research, which is based on plant physiological, morphological, and phenological responses to drought, a meta-analysis. Human activities are responsible for ongoing rapid climate change, and it is predicted that climate change will alter global precipitation regimes. Plants can respond to changes in precipitation via a diversity of trait strategies, and therefore we observe different plant responses to drought. And the specific question we seek to answer is, are drought responses mediated by functional traits and habitat of origin? We hypothesize that drought will affect the morphology, physiology, and phenology of plants, and we expect that these effects will vary across habitat types and differ in their aridity levels. And the method we use is a meta-analysis to investigate the effects of drought on plants via functional traits. Our preliminary results show that responses associated with morphological and physiological traits are similar in magnitude, implying that both types of traits are likely to exhibit substantial changes with drought conditions, and also that only plants in savannas and arid environment exhibited significant changes in response to drought. However, these are preliminary results and are expected to change as more data is included.
Hello, my name is Teresa Osorio, and my research is titled Exploring the Role of Mitochondrial Endopolyphosphatases, MitoPPN, in Mammalian Bioenergetics. Poly-P is an ancient biopolymer that is well preserved throughout evolution and present in all steady organisms. In mammals, it shows a high co-localization with mitochondria and has been demonstrated to be involved in the homeostasis of key processes within the organelle, including mitochondrial bioenergetics. However, the exact extent of the effects of poly-P on the regulation of cellular bioenergetics, as well as the mechanisms underlying these effects, have remained poorly understood. Here, using SHSY5Y mammalian cells under wild type and mitoPPN, cells expressing the PPN enzyme in the mitochondria conditions, we showed that depletion of poly-P within mitochondria does not increase oxidative stress conditions. This is characterized by a decrease in basal antioxidant protein levels, which are critical for detoxifying free radicals. Moreover, we demonstrate a significant effect of mitochondrial poly-P on the regulation of global cellular bioenergetics in these cells.
Hello, my name is Sashoya Duggan.
And I'm Alexis Winters, and we will be presenting the phylogenetic analysis of adenoviruses found in Anolis sagrii and Podarcis siculus. While we worked separately on different projects, each in different stages of their completion, they both aimed to further the knowledge of squamate adenoviruses.
Adenoviruses, also known as ADVs, are non-enveloped DNA-dependent viruses able to encode their own replication factors when entering a host. They commonly infect a wide array of species, each affected in different ways. Most reptilian cases are caused by ADVs within the adenovirus genus, yet more recently, 2 individuals of Anolis sagrii from the islands of Staniel Key were found to be infected with ADVs more closely related to the mast adenovirus genus, previously known to primarily infect mammals.
In addition to this, little is known about ADV in a more wild population of lizards such as Podarcis cichlids, which have never been investigated for their history of adenovirus affliction.
In hopes of better understanding the origins of these novel ADVs from Staniel Key and its effects on Podarcis cichlids, we have continued ADV research with these lizards species in our lab. This process included a series of PCR and nested PCR tests to amplify adenovirus in these individuals and sequencing of the successful amplifications. Our results indicate that adenovirus is present in our samples. The positive PCR results suggest this inference. Of the samples, Anolis sagrii positives were recorded and able to move on to Sanger sequencing, while the Podarcis cichlids samples read positive in the first run of PCR analysis, but not the nested.
Going forward, we plan on rerunning the Podarcis cichlids individuals PCR testing using new reagents to solidify our positive results, and we plan on generating new phylogenies based on these positive reads we got from Anolis sagrii.